All right, so chapter 7, part 3, we're going to talk about the dark reaction. So we already talked about the light reaction. And so with the light reaction, we get ATP and NADPH out of that. It's then going to be, those products are going to be required for the dark reaction. So this one we call the light reaction. So because this one doesn't use, since this one here does not use light, we called it the dark reaction. But the problem was that it can occur for a short period of time without sunlight. But once you run out of NADPH and ATP, you can't continue that process. So then they tried to give it a better, more appropriate name. So they said, how about the light independent reaction? Because it's independent of light, but that's not true. It is dependent on light. It needs ATP and NADPH. So then we had someone who actually worked the steps of this process out, Dr. Kelvin. So we called it the Kelvin cycle. So we frequently will refer to this as either the dark reaction, the light independent reaction, or the Kelvin cycle, or I'm going to throw another term in there, the C3 cycle, which I'll get to at the end of today's presentation. So this is the Kelvin cycle. I am going to go over this and basically on the board, and I have a video of me doing that. And what we really want to look at when we watch this process is you need to follow the carbons. So these are all the carbons, 1, 2, 3, 4, 5. We have 5 carbons here. This is a 5-carbon sugar, RUBP. It is then going to use this enzyme, Rubisco. Rubisco is going to take RUBP and our 1 carbon, right? 1 carbon, and put it together into, and they don't really show this. I'm going to put it up here. 3, 4, 5. A 6 carbon. A 6 carbon. Oh, and I don't think you guys can see that where it's at. Let me put it somewhere else. So over here, so I'm going to write it down here. It should be up there. Is a 6 carbon, 2, 4, 5, 6 molecule. And that 6 carbon molecule is a transition molecule. And what happens is it splits in half. And so notice here I have 3 carbons and there are 2 phosphates, right? So 1 here, 1 here. So we would have the phosphate here and the phosphate here. And so we split it in half. And then we're going to add in some energy from the light reaction. We're going to throw some hydrogens on it, right? Because we have carbon here and we have oxygen here. Well, remember our formula for carbohydrates are CHO, CHO. We're missing that hydrogen. And who's going to bring that? Right there, NADPH. So we're now turning that inorganic molecule into an organic molecule. We're fixing it. So when we talk about fixing, it means that we're making it work. And so we're taking something that's inorganic and making it organic. And that is what's happening in that first phase as Rubisco grabs that molecule. And then we complete the process while we reduce it in phase 2. Now I'm not going to balance all these. I'll balance them on the video, all the numbers and such, but you just need to know that you don't just have one of these molecules. You have 6, you have 6, 6. We have our G3P. We saw G3P in glycolysis. We have 6 of those molecules. 1 is going to exit. So we have 5 left. Going to add again some more ATP energy from the light reaction, and we regenerate and start all over again with RUBP. All right. So let's see this step by step here. So again, RUBP, we have 1, 2, 3, 4, 5 carbons. We're going to add our 6. So we have 3 of those molecules and 3 of those molecules. So technically I have 3 times 1, 2, 3, 4, 5. I have 15 carbons. And I'm adding, notice I have 3 molecules of carbon dioxide. So I'm adding 3 carbons. So now I'm going to have a total of 18 carbons. And if I look at this molecule right here, I do, right? 6 times 3 gives me 18. I got ahead of myself there. All right, so we added 3 carbons. We fixed 3 carbons from the atmosphere. And now we're going to add some ATP. We're going to add our hydrogen and electrons. We're going to reduce it. We produce our G3P. 5, we have 6 of those molecules. 5 are going to stay in the cycle. 1 is going to exit. Now it is that one that exits right here. Wow, I can't do both. All right, the one right here that is going to make those other compounds. So it's going to make glucose. So we're going to need 2 G3Ps to make glucose. We're also going to use that G3P to be our glycerol backbone for lipids, phospholipids, triglycerides. We're also going to use those glucose molecules, right, to make things like starch or cellulose. And then we also can use those 3 carbons as our skeletal or framework for our amino acids. So we have 6, 1 exits, that leaves us 5. So basically I'm losing my 3 carbons, right? I put in 3 carbons. Whoops. I put in 3 carbons. And then I took out 3 carbons. So our law of conservation of matter and energy is still at play here. All right. We add a little bit more ATP and we're back to RUBP. Don't forget our enzyme Rubisco here. Okay. So the fate of G3P. So like I said, so G3P can go in a couple different places. We can go and make glucose. The glucose then can make fructose. We have an enzyme that will convert glucose to fructose. And then we can put a glucose and a fructose molecule to make a disaccharide in the form of sucrose. And that is actually how that sugar is transported around the plant. We can also then take the glucose and we can make our starch and our cellulose, our cell walls. So we have our storage and our structural support. We can then, like I said, go to lipid synthesis, make different lipids. We could also then go and make the different amino acids that we will need so that the plant can make proteins. Now, there are some adaptations plants have made due to the environment in which they are able to inhabit. So when we talk about C3 plants, we're talking about the Calvin cycle because as you notice, and we can go back and you can look, many of the intermediates in the Calvin cycle have 3 carbons. There is a problem though that plants have with just normal photosynthesis, and that is when it is hot and dry outside, they're going to lose a lot of water. And if we lose a lot of water, then it's going to cause the stomata to close. And now that our stomata is closed, we can't get gas exchange. And so gas exchange comes to a halt. And this is going to lead to a situation called photorespiration. Because you got to think about it now. In that spongy, the spaces in between the spongy mesophyll cells, which is those pockets we have for the gas to come in and out, the oxygen can't get out, the CO2 can't get in. So now we're going to, as we use up our CO2 concentration will go down inside the leaf and our oxygen is going to go up. Now, what we really want is we really want this process here where CO2 binds to Rubisco, our enzyme, and everything goes nice like we want, and we get out our sugar. Yay, that worked perfect, right? We have our sugar. So once we have, and I'm fighting with that darn pen again, once we have our sugar, okay, everything's good. We used all the energy from the light reaction. Wow, I had this thing charging all night. So we have the energy from the light reaction. You'll see me toss a pen here. And so, and then we had all the energy here from the light reaction. All right, I'm just gonna move on here without my pen. And then versus photorespiration. So when we look at photorespiration, this is where we are going to be taking in. So when we look at that other circle, which I can't really point out right now. So when we look at that other circle right here, it's taking in oxygen and it's binding with Rubisco. And so when we bind with, and we think about it, photorespiration, photosynthesis, as we take in CO2, we give off oxygen. Respiration, cellular respiration, right? We took in oxygen, we gave off CO2. So this is where photosynthesis is respiring. It's taking in the oxygen and giving off the CO2. So if you notice in the circle, instead of making sugar, we're kicking out CO2. So we're using that energy, those products from the light reaction, but we're not making any food. So it's counterproductive. It's a waste of energy and resources. You can think of oxygen as being a competitor, a competitive inhibitor. We talked about this. So what we know about Rubisco is Rubisco has been around longer than free oxygen. So when we talk about our planet's history, right? Life started in the water. We started with photosynthesis in the water. Once the water became saturated with oxygen, it leaked into our atmosphere. We did not have free oxygen in our atmosphere, right? So once it took millions of years of photosynthesis to get oxygen into our atmosphere like we do today. But in the beginning, there was no free oxygen. So Rubisco could work just fine with all that CO2 and it didn't have to worry about oxygen. As oxygen levels increase, we now have a problem because it happens to fit in the active site of Rubisco and it blocks out CO2. So it is a competitive inhibitor. So this is a remnant of, of evolution here. So we see that oxygen will bind in a normal situation will bind with Rubisco about 1 every 3 turns. All right, so we do not have perfect efficiency with photosynthesis because of photorespiration. Now, there are plants that live in an environment where they don't close their stomata and have free gas exchange, not a problem. But in places where it gets really hot and dry, it's a problem. And so plants have come up with 2 mechanisms in order to prevent this problem. So what they're going to do is the C4 plants are going to actually store, the C4 plants are going to actually store carbon dioxide in their mesophyll cells, and they're going to store it as a C4, so it has 4 carbon atoms in it. So this is where we get the C4 from. And so when it's hot and they start losing water, the stomata is going to close midday during the hottest time to prevent water loss. And that's okay because it's got CO2 stored for when we have low amounts of carbon dioxide, right? So when we're not able to get that carbon dioxide from outside, it's got it stored so it can release it and then use that to help reduce the photorespiration and to continue to make food. Now, cam plants, different story. Now, cam plants live in really, like these are your cacti, orchids, pineapples are an example of cam plants. They live in very, very hot and dry environments. And so, or at least environments where water loss is a serious issue. And so what's gonna happen here is that cam plants only open their stomata at night. So they open their stomata at night, they collect the CO2 and store it. They store it also as a C4. It's very similar. We don't, when we talk about evolution, we don't have large changes. We just have small tweaks, right? And those small tweaks allow certain environments, certain organisms to be more successful in that habitat. So this small tweak allowed plants to live in the desert. So what they do is they only open their stomata at night, they collect their CO2, and then once daylight comes, right now we have our ATP and our NADPH, they'll use that CO2 that they stored because their stomata is closed during the day. Now these plants typically grow very slowly because, again, they can only collect so much CO2. They don't have mass storage containers for CO2. So these plants, again, this is what allows them to live in those very hot and dry environments. So we have C3, C4, and CAM plants. Those are all modifications, again, the C4 and CAM to reduce photorespiration. All right, so again, that's the end of the Calvin cycle. I have several videos, and I also have some videos of myself going through that process, and hopefully you'll find that those are helpful. All right. If again, as always, you have any questions, feel free to send me an email. Thanks. Bye.